Hey guys, it is Professor Parkinson, and I'm going to do a quick overview of histology. Um, the tissue chapter is coming up soon in lecture, but we have to go ahead and learn about tissues and histology in our lab section. The histology section is very large. We will cover chapter 4, 5, and 6 in our lab manuals, and we will start doing the experiments next week. But since we don't have a Monday lab next week, I want to go ahead and run over this with you guys so you can begin to study because our first practical exam is coming up shortly. So the components of a tissue, we have cells. All tissues contain at least one type of cell, but there is a large variety. We also have a matrix, or the extracellular materials. We have ground substance, which would be a gel or fluid base, and fibrous proteins. Epithelial tissue. So epithelial tissue has the distinct physical characteristics. They have large numbers of cell and little visible ground substance. They are avascular, meaning they lack blood vessels. A, we should know, means without. So avascular. They do have attachment to connective tissue by a thin, complex basement membrane, and we will be able to identify the basement membranes. This anchors tissues and regulates material exchange. It also has a polarity, so there are distinctive sides to the cell. We have apical. They are superior or free surface. They face away from the basement membrane. Or basal, which have inferior surface and faces towards basement membranes. So when we talk about cell shape, we have squamous, which are thin and flat or irregular edges. Cuboidal, they are tall as they are wide, and they are roughly square-shaped. They may not always be exactly square-shaped, or they may appear round. Columnar, they are taller than they are wide, and they can be tall and narrow. Simple versus stratified tissues. So simple is only one layers of cells. They are low stress areas, and all cells touch the basement membrane. You can see here is our basement membrane, and then the cells. So stratified, they can have between 2 and 20 layers. They are high stress areas, and they only the deepest layer contacts the basement membrane. So you can look here at the basement membrane again, but when we're looking at cells and trying to identify their shape, we need to understand that we're not looking at just this first layer. We need to look at the top layer to be able to identify what shape this is. Down here we might think, oh, these are cuboidal. But in actuality, if you look up at the top layer, they are squamous. So in pseudo-stratified columnar cells, some cells are shorter and some are taller, but they do all make contact with the basement layer. Pseudo meaning they are fakely stratified. All right. Simple squamous epithelium. We are looking at this, the sample from the top down. So we are looking down onto it from the top layer. The structure is a single layer of cells. The cells are squamous, irregular, flattened. The nuclei are flattened or rounded. This is the top view structure. Here you cannot see the basement membrane. So when trying to remember this and identify them, they do kind of look like a fish or reptile scales. And we will only look at epithelial tissue shown from the top view. So make sure that you are paying attention to that. Also, please do not pay attention or memorize the types of tissue based on the color of the tissues, because every labs use different stains, and we don't want to think, oh, this is a purple tissue, because we could use a blue or a pink stain. Simple squamous epithelium. This is a side view. So this is mostly how we will look at all other tissue. This structure is a single layer of cells. The cells are squamous, so they are regular and flattened. The nuclei are flattened or rounded. They have a thin cytoplasm. Cells kind of look like fried eggs. They are the thinnest of tissues, and they typically can only spot nuclei projecting from the edges, like here. You can see the basement membrane here. All right, simple cuboidal epithelium. So the structure is a single layer of cells. Cells are rounded in shape, and they are arranged in neat rows. And also a lumen or lumens may be present. So when you're trying to remember this, for identification, slides often show cells arranged into tubes or rows like this. And nuclei are round, and it will only be shown from the side. All right, simple columnar epithelium. This structure is a single layer of cells. Cells are taller than they are wide. The nuclei are oval and vertical. They may possess microvilli, which are up here. You can barely see them. And they also can have goblet cells. So remember that the nuclei are nearer to the basal end of the cell. The nuclei they're around similar depths. And the microvilli it kind of appears a fuzzy border. So you're not going to see the individual strands of the microvilli. So the goblet cells are wide on top and narrow on bottom, and they're filled with secretory vesicles. All right, pseudostratified columnar epithelium. So this is pseudostratified. So if you look here, it looks like there's multiple cells, but in actuality, it's just one. So the nuclei are at different heights, and the cells are different heights, but there's only one single layer, and you can also see the cilia. So you're going to see the individual hairs of the cilia on this type of tissue. So again, pseudo means false, because this is actually a simple tissue. It is not stratified. All cells touch the basement membrane down here. And this is the only ciliated tissue that we will see and use. Stratified squamous epithelium. The structure has multiple layers of cells. The basal cells look more cuboidal than columnar, but this is why we have to make sure we're looking at the top layer. So the superior cells are still alive up here. There's one of two, this is only one of two stratified tissues, and the tissues are named for the shape of the apical cell, so up at the top. Stratified squamous epithelium, this is keratinized. The one previously is unkeratinized. So the structure here is multiple layers of cells, and the basal cells look more cuboidal. And the cells flatten as you move superiorly. So the superior cells lose the nuclei and they become dead. So these are one of the two stratified tissues that are more visible strata or layers. You can see all the layers. And the wavy edges where the epithelium meets the connective tissue, such as here. This is a very thick tissue. So transitional epithelium, the structure has multiple layers with rounded cells, you can see. They are often shown in cross-sectional of the uriner, so a rounded tube here. They surround hollow lumen, so open space. The superior cells remain round and not flat. And there's transitions between two states. So if the tissue is relaxed, it can be more rounded. And then when it's stretched, they can flatten out like here. Transitional epithelium will not look like any other tissue. All right, I'm going to pause here at connective, and then we'll start a different video.